So, ladies and gentlemen, I think we should get started. In the interest of time, we're honored to welcome Dr. Jean-Luc Fréquin, who's the CEO of Spectradyne. And Dr. Fréquin will be discussing the new Spectradyne technology, the ARC particle analyzer, today. If you have questions, ladies and gentlemen, you can raise your hand. So at the end of the talk, we can unmute you so you can ask in real time the question. Alternatively, you could type it into the Q&A box and we can address them, questions in the order received. So without any further ado, ladies and gentlemen, let's welcome Dr. Fréquin to initiate the presentation. Welcome, Dr. Fréquin.
Thank you. Good morning, everybody, or afternoon, depending on where you are. Thanks for joining us today. I understand your time is precious, so hopefully we can fill it with exciting stuff for you. Today's webinar is entitled New Insights from Spectradyne's ARC Particle Analyzer. And my goal for the webinar is to show you some of the cool things that people are doing with the ARC, which we launched in the fall, and to answer any questions you might have about the technology. So if you're new to Spectradyne, if you're not familiar with us, we make instrumentation for accurate nanoparticle analysis. And our goal is to reshape the discovery, development, and production of the way of modern therapeutics, the way new drugs are being made and discovered. Today in the webinar, we'll talk about why it's important to measure nanoparticles accurately, talk about how it's normally done currently, and then introduce our ARC particle analyzer. I'll give you a brief overview of how it works before we dive into the measurement examples that you're here to see. So, as we all know, nanoparticle applications are growing rapidly, right? Probably everybody's had a nanoparticle nanomedicine vaccine. There are gene therapies based on nanoparticles now, and new diagnostics and fundamental biology being developed all the time. The characterization of these particles are critical because at this size scale, the size and the concentration of the nanoparticles directly impact their performance very strongly. So, for example, if you think about it, the size and concentration of the particles in the sample really determine the amount of material, the amount of drug in the sample, and that's the dose directly, right? The size of the particles also impacts the diffusion very strongly of the particles in a recipient of a therapeutic, and so that's the bioavailability of the drug. And ultimately, these add up to impacting the safety and efficacy of the therapeutic also. Critically now, these drugs are being made in very complex samples. So they're extracted from biofluids, they're grown in cell culture media, or from extracted from cell lysates. So these are complex soups of particles and mixtures of things, proteins and other reagents. And so it's important, if we're going to measure the concentration of just of the active ingredient, for example, that we be able to identify exactly what particles we're measuring in the sample. So phenotype identification is essential. So essentially, it's size, concentration, and phenotype. These are key parameters for developing effective nanoparticle-based therapeutics, as well as, I'll show in this next example, any sort of new biological discoveries. So let me illustrate this with an example, and this is kind of a canonical example, independent of the application which you're using nanoparticles for. But we're imagining we're scientists and we're trying to compare two formulations that are nanoparticle based. So we've got formulations A and B, and typically the way you evaluate these is you put them on a test system. Maybe it's cell culture media, or maybe it's a live animal model, and we try to measure the effect of these two formulations and compare them, right? So in this simple example, formulation A did not change very many of the cells to be fluorescent. Formulation B changed a lot of the cells to be fluorescent, and we're detecting that shift by, say, flow cytometry, schematically here, right? But not one scientist in the virtual room that we're in right now would do this experiment without first controlling for the amount of material that we put on the test system, right? It's critical that you put the same amounts of A and B onto the cells in order to compare them properly, right? So accurate size and concentration are critical for doing good science of nanoparticle-based therapeutics or new biological discoveries. One important point here is that there are a lot of tools for evaluating the outcome, right? These are usually in cells or animal phenotypes or lifespan measurements or something. And there are a lot of good tools like flow cytometry for evaluating the fluorescence activation of cells, for example. But there are not a lot of good tools for normalizing and quantifying the products that affect that change. Current workflows typically require multiple CapEx instruments. So I'm showing you a few here. Dynamic light scattering is a workhorse in the nanomedicine applications, even though it doesn't give a lot of information about the sample. There are surface immobilization techniques that require putting the sample on a microarray. There's flow cytometry, of course, which can be, it's a very complex technology. There's nanoparticle tracking analysis. And for virus quantification, a big workhorse still is really plaque assays, and these are tissue culture based. They can take days, right, to do. So many different instruments required in order to quantify nanoparticles. And I'll point out here, every one of these technologies is light based, right? So this is light scattering. This is interferometric imaging. This is light scattering. This is light scattering, and well, this is just microscope imaging, right? But because they're light-based instruments and technologies, they're indirectly measuring the nanoparticle size, right? They're not well suited for measuring particles that are smaller than the wavelength of light, right? Used to image them. As a result, it takes a lot of work to sort of to synthesize the results from all these measurements and try to make a complete picture of what's really going on in the sample. So the data analysis is complex. You have to understand the limitations of all these technologies. And ultimately, frequently, these results can still be misleading, actually, and tell you something that's not true about your sample. So there's a critical need for direct and accurate measurements of nanoparticle size, concentration, and phenotype in this industry. Spectradyne's ARC particle analyzer does exactly that. It makes accurate concentration and size measurements of particles at the nanoscale, as small as 50 nanometers in diameter. And I'll get to this in a minute, but it's not it's not based on the particle detection is not an optical method. And so it means we're independent of any kind of material. So whereas the light scattering technique, you would need to know things about the optical properties of the particles that you're trying to measure. In our system, you don't need to do that. It works for any material. The ARC also makes quantitative single particle fluorescence measurements. So that's how we do the phenotyping and the subpopulation analysis in very complex samples. And all this happens in a single instrument that's fast, easy to use, and compatible with the flow cytometry protocols and reagents that are so powerful and have been developed over the last few decades for flow cytometry. It's a unique combination of two techniques. So we use microfluidic resistive pulse sensing, or MRPS, which is an electrical particle detection method. As I mentioned, it's non-optical, and that means that we can access sizes more accurately below the wavelength of detection, wavelength of light used in the light scattering techniques. We're also making quantitative fluorescence measurements. So we default to reporting NIST traceable units for brightness, for fluorescence intensity in our system. We can detect up to three fluorescence colors. These are user configurable very easily. And I'll show you later, the ARC is very sensitive in the fluorescence detection channels, comparable to or better than most flow cytometers that are on the market right now. In addition, the technology leverages microfluidics, and this has many advantages, but in particular, for most people, this is exciting. It only takes 3 microliters of a sample for analysis. So the magic happens inside this microfluidic cartridge. It's about a ½ inch on the side. And what we do with the electrical sensing technique is we flow the sample through constriction in the fluid flow, okay? We apply an electrical bias across here so that every time a single particle goes through the constriction, we get an electrical signal that tells us two things. First, the size of the particle, and second, how fast it went through. And that can tell us the amount of sample we measured and therefore the concentration of particles in the sample. So this is the most direct method of measuring size and concentration at the nanoscale that's available. And actually it's based on the Coulter principle, right? It's just a nanoscale version of the Coulter principle, which has been a gold standard for measuring large particles in cells for decades, right? It's the foundation of Beckman-Coulter. Now in the arc, what we're doing is we're exciting the particles with a laser at the exact moment that we're measuring them electrically. And we're collecting the fluorescence light that's emitted from the particles when they fluoresce, if they fluoresce. And so simultaneously with the electrical signal, we're measuring the fluorescence in up to 3 optical channels to get the phenotype or the measure of the payload, which we'll talk about in a bit, by fluorescence. So this is how we get the specificity. This cartridge is a key part of our technology, so let me show you a little bit more about it. So it's made with cutting-edge microfluidics. These are nanofabricated cartridges. They include a couple of key features. So what I'm showing here in panel A is an input filter at the input of the cartridge. What this does is it excludes larger particles from the sample that would otherwise block the constriction where it's being measured, where particles are being measured. And that allows people to, users to measure very complex samples like cell culture media, for example, directly on the chip. We have very precise flow control inside the cartridge. And in image B, what I'm showing you here is particles, fluorescent particles transiting the sensing constriction. And you can see the beautiful laminar flow that is taking place inside the cartridge. And the whole cartridge is designed with an electrical architecture also. So that's what's shown here. This is basically just a cool picture of an electrical element inside the cartridge that allows very fast measurements. So technicalities aside, what this means for the user is you can measure very complex samples on the cartridges. The cartridges are nanofabricated, they're consistent in dimensions, and so we can pre-calibrate them at the factory. And as I mentioned, because we're in microfluidic system, it's only a very small sample volume required for analysis, just a few microliters. Basically, as small as you can pipette is good enough. So this technology has main key impact areas in nanomedicine. So for example, LNP quantification, where the ARC will deliver safer, more effective drugs and faster development times. In virus and gene therapy applications, it will enable higher quality vectors. So we'll be able to have a sense for, or be able to measure what fraction of your lentivirus carries the genomic material for the transfections. And ultimately more efficient processes there. And in extracellular vesicles research, this is earlier stage generally, but so it will allow for better quantification, better controlled science, and ultimately new discoveries this way. So let me get into a couple of examples then to show you some of the exciting things that we can do with the ARC, starting with nanomedicine applications. So here we're going to look at lipid nanoparticle formulations and characterize their payload encapsulated of RNA in the samples. So what I'm showing you here are measurements of 3 different LNP formulations. These were made by the Nanotechnology Characterization Lab in Frederick, Maryland. Thank you, Jeff. These 3 samples were made using a commercial LNP production tool, a benchtop manufacturing system for making RNA encapsulated LNPs. And there are 3 different samples here. So the red sample are LNPs that were produced with no RNA in them. And then there were 2 formulations made with RNA. One was a formulation that resulted in a low encapsulation efficiency of RNA, and the blue one is the high encapsulation efficiency LNPs. And so the way, just as a side note, the the only way that people generally have to evaluate how much RNA gets taken up into the LNPs is to do a bulk analysis, like a ribogreen assay of the free RNA in solution after the encapsulation step. And so you know how much RNA went in, you know how much is left over in solution that did not get encapsulated from the ribogreen assay, and then we assume that the rest of the RNA goes into particles, but we don't know anything about the uptake, the size distribution of the particles that were uptaken, and how much RNA is in each particle, right? That's where the ARC comes in here. So what this plot is showing us is the real particle diameter on the x-axis as measured by MRPS. So this is our electrical particle size and concentration measurement. And on the y-axis now is the brightness in the FITC channel of individual particle events, okay? And what we did to get these particles to fluoresce is we stained them very simply with the cyber gold. It's an intercalating nucleic acid stain, goes into the LNPs and lights up the ones that have RNA inside them, right? And so if we look at the red sample, the no RNA, the empty LNP sample, there's very little fluorescence as expected, right? Then the low encapsulation efficiency ones are shown in green, and so there's definitely more fluorescent particles in here in this sample, right? And in the blue are the high encapsulation efficiency LNPs, and we can look just qualitatively from this graph and see that there are more brighter particles in the high encapsulation efficiency LNP formulation than in the low encapsulation efficiency formulation. So that's a good start, and we can be, we're already quantitative. Look, these units are in ERF units. These are translatable units of absolute brightness. So if a particle shows up at 1,000 units of FITC, it means this particle is as bright as 1,000 free-floating FITC molecules in solution, right? This is a NIST standard. So these are not arbitrary units that you might commonly see from a flow cytometer in terms of voltage on the detector or anything like that. These are calibrated. All right, so we can be more quantitative than this though. So, right, so the particles are brighter, that means there's more RNA in them. But let's look at the distribution. So now this plot is showing us concentration on the y-axis in real units, particles per mil. And then on the x-axis, we're showing the brightness now. So this is the same scale as here, but now we're showing the concentration as a function of brightness. So again, the empty LNPs, there are very few particles above threshold, right? It's not a fluorescent sample because the particles are empty. And then we can see a real difference between the low and the high encapsulation efficiency LNPs here. The high encapsulation efficiency LNPs have quantitatively more, higher concentration of brighter particles than the low encapsulation efficiency ones. So this is a unique insight now into the encapsulation process of LNPs for this very common application now, right, for nanomedicine. Where before we could only do bulk assays to try and infer how much payload was being incorporated in the particles. Now we can actually measure that on a particle by particle basis. Okay, so the second example I would like to talk about is about extracellular vesicles. And so here what we did was a subpopulation analysis. We took two samples of EVs, extracellular vesicles. One of them expresses GFP on intraluminally, and the other population they're non-fluorescent, just regular EVs. And we put in the fluorescent ones at a low fraction of the sample. So if we just look at the total particle size distribution, this is our MRPS technique, right? So what we see is a broad size distribution of particles with approximately a power law size distribution that's very common for EV sample preps. And this is what you would get from our Gen 1 technology. It's already more accurate concentration of particles in the sample than other techniques at the nanoscale. but it doesn't tell us about the composition of the sample, what kinds of particles are in here very much, right? It just measures all the particles. But now on the ARC, we're measuring fluorescence from every particle. And so here's an example where I wanted to show you a little bit of the raw data to give you a flavor of what it looks like. These three curves are showing us the raw data coming in measured for MRPS, which is the total particle measurement, the electrical method in blue, and then the signals in two of our fluorescent channels, the green and the yellow channels. So what we see here, I've highlighted with an arrow, is individual particle being detected by MRPS. Okay, so this is the electrical measurement of size and concentration of this particle. And then simultaneously with the MRPS signal, we're detecting fluorescence in the green FITC channel as well as the yellow PE channel. So now we can get into more nuanced subpopulation analysis of this sample. So before we had the total particle size distribution, now we can look at the size distribution of just the fluorescent FITC positive particles in the sample. And now we can identify the subpopulation of samples or of particles in the sample, and we can be quantitative over any size range here too. So whereas the total particle size distribution showed on this size range concentration of about 1 × 10 particles per mil, the fluorescent subpopulation is a factor of 10 lower, only 8% of the particles here. And that's how we mix the sample up. And this highlights that in a single measurement on the ARC, we're able to assess the size of this subpopulation, their concentration, as well as their fraction of, of this phenotype in the sample. So this is like a single measurement of purity as well in this measurement. Okay, so if this is your active ingredient in your science experiment, this allows you to accurately control for the dose on the different, in the different test systems, and really get an accurate comparison of your different formulations as you're doing your science. Okay, so those EVs were just, they were, genetically engineered in order to express GFP, right? But what about immunostaining? So let me show you an example of that. So here we took EVs that were isolated using an affinity bead based method and eluted from the capture beads. And we stained them for CD81, which is a human tetraspanin marker. We stain them in two ways. One is with a PE labeled antibody for CD81, and also with a PE labeled isotype control, just an IgG, right? So you can see that the particle size distribution is not significantly impacted by the antibody staining. And so let's look at the fluorescence now. So comparing the two examples, the two samples results in this clear difference here. So this plot is showing us, again, brightness on the y-axis and particle diameter on the x-axis. And now we're looking at the PE channel, and you can see the limit of detection here is about 2.5 PE. So again, these are real translatable units, which means particle here is as bright as 2.5 PE molecules free floating in solution, right? So what we see from this plot now is that the isotype control is not binding to the EVs, right? It's not resulting in fluorescent particles. And so we can conclude that the staining is specific, and we can be quantitative over the brightness of all of these particles as we did before. So this plot here is now showing us the concentration on the y-axis versus the brightness on the x-axis is in units of PE molecules, right? And so this clearly shows us that the immunostaining is specific for CD81. And we can say that the median brightness across this distribution is about 6 or 7 molecules of PE per particle. So that allows you to get at, well, how many copies of CD81 are expressed on these particles on average and as a function of size, right? And we know that from other of our collaborators' research that's to come, that the brightness here is proportional to the surface area of the particles. So if you plot this versus surface area, it shows up linearly, which is expected, right? Because these markers are on the surface of the particles. Also, as before, we can plot then the concentration of the subpopulation as a function of size. So here we're showing the total population of particles in dark blue, as well as the distribution of sizes of particles that are PE fluorescent positive, and be quantitative about the amount of fluorescent particles in the sample this way. So these are quantitative measurements with standard default units or default standard units, right? So the concentrations in particles per mil, diameters in nanometers, and fluorescence is in real translatable units of MESF or ERF units, depending on the fluorochromes being used. Okay, so as the last example, I would like to show you an example of virus measurements. This is relevant for gene therapy applications, and an illustration of how quickly we can measure the virus titer in a sample, specifically even in a very complex sample. Okay, so what we did here was we took human adenovirus 5 and we mixed it with a bunch of EVs, right, that had a broad size distribution. Okay, and we stained all these samples with cyber gold again. So in the case of the adenovirus, the cyber gold intercalates into the genome in the virus and lights them up fluorescently, whereas the EVs don't carry significant nucleic acid in the sample to be detected, right? So what I'm showing here in this plot is the MRPS measurements, so the total particles in each of the samples. So in blue are just the EVs, in green are just the adenovirus by themselves, and then the red is the mixture. And you can see just right away that these distributions add together, right? Which makes sense. If you mix them together, you should get a superposition of the two distributions of particle sizes. And I challenge you to do this accurately on any other technology out there. So this is unique to MRPS. It's excellent precision and concentration and size. So, but now on the ARC, we have more than just the electrical measurements to do. So, so let's look at the fluorescence from the same sample. So now what I'm plotting here are the brightness, again, in real FITC units. So these are cyber gold is essentially a green dye. So this is brightness on the y-axis and real particle diameter on the x-axis down to 65 nanometers or so. We see this really interesting clustering of events here. So adenovirus is about 85, 90 nanometers. That's where this peak shows up, and that's where this fluorescent population shows up, okay? And then there are other populations here, other clusters. And if you think about it, a virus dimer should be twice as bright as a virus monomer. And that's exactly where this population shows up. And it should be twice the volume of a virus monomer also. And that's where this effective spherical diameter shows up. So these really are virus dimers. They're twice as bright as the monomers and twice the volume of the monomer. And then if you squint your eyes, I didn't put arrows on these, but there's a cluster here of trimers and even quadrimers also visible at the correct brightnesses, relative brightnesses, and relative effective spherical diameters also. So this is a very powerful technique, high resolution measurement. And with the fluorescence here, we've been able to isolate just the virus particles from all this non-fluorescent EV mixture in the background, right? And we can be quantitative about how bright these particles are. In this plot, we're showing the EVs on their own brightness distribution in blue, and then the brightness distribution of the virus. And you can see these humps from the dimers and trimers also in here. And so we can say that, well, the virus, the mode of the virus brightness is around 80 units of FITC. So it's about as bright as 80 free-floating molecules of Phe in solution. Okay, let's talk about how low we can go with this technology. So a little bit about the sensitivity of the ARC particle analyzer. And I want to explain or describe to you how we do the calibration of the brightness on our system, and how it is that we can report standard units for brightness. Okay, so what we did was we took a pair of polystyrene beads, and these have they're solid dyed beads with a YG fluorophore. Okay, and there are two different diameters, 180 nanometers and 290 nanometer diameters. And these particles are, they're sort of unknown brightness until you cross-calibrate them on regular flow cytometers. So where there are MESF standards, NIST traceable MESF standards to compare their brightness to. Okay, so what we did with one of our collaborators was we sent these beads to them. They did a one-time brightness calibration relative to NIST standards. Okay, so these are compared to the 8 micron MESF standard beads. And this was done on a conventional flow cytometer, and they assigned brightnesses to both of these bead sizes relative to those MESF standards. Okay, so, and importantly, those measurements were done with a known excitation wavelength and detection bands on the fluorescence detection channels. So what these collaborators found was that in the Phe channel, the 180 YGs were as bright as 964 free-floating molecules of Phe, and in PE, they were as bright as 20 free-floating molecules of PE in solution. Okay, the 290 YG beads were brighter. They're bigger beads and they're solid dyed, so there's more fluorochrome in there. And those were assigned brightnesses of about 7,500 in the Phe channel and 174 PE in the PE channel. Okay, so now we know absolutely how bright these two particles are, and we run them on the ARC, which actually this is already done here. But let's look at the brightness distributions of these samples. Okay, so first focusing on the Phe channel. We're showing the full measurements here. So there is a peak in the distribution from the 290 YG beads and the 180 YG beads. And then down here is our noise level. Okay, and then we're going to come to that because we want to measure the noise level, the detection limit here. So we scaled this axis so that the 290s matched the assigned value from the cross calibration. Okay, and then we looked at the 180s. We saw the 180s match the independent brightness assignment from the MESF cross calibration also. So these are coming out 964 or 1000, and when the 290s are aligned with their assignment also. We can do the same thing in the PE channel. So we align the 290s with the assigned value. We confirm that the 180s match the independently assigned brightness. And now we're set up to look at where the noise floor is for both of these channels. So what we can see is that the noise starts kicking up around 20 molecules of Phe. So that means that we should be able to detect a particle that is as bright as 20 free-floating molecules of Phe in solution. And in the PE channel, PE is a much brighter fluorochrome, and so here the sensitivity limit is, it's higher sensitivity, lower detection limit for PE. We're in the single molecule of PE range of sensitivity here. So these are at least or better sensitivities to most flow cytometers that are currently on the market right now. All right, so at this point, I would refer you to our website to learn more. So we have a lot of videos and we're constantly putting up new information. We have application notes about each of the applications I talked about today. We've got a product page about the ARC. And actually, we have a new website in the last few months. I encourage you to go check it out. It's fun and exciting. We've got a recording of the product launch event from last fall, which features good speakers from the nanoparticle analysis field. We've got virtual demos and lots to learn on our website about this technology. And I would also like to put in a plug for us shamelessly winning the technology showcase at the CYTO conference. This is the International Society for the Advancement of Cytometry. At their annual conference, which took place last month in Montreal, they had a technology competition where up-and-coming technologies get to make their pitch, and we were voted by a panel of experts to win. So we're very proud about that. And we encourage you to come meet us soon at a trade show in person or contact our sales team to learn more. This is our next one, probably in July. We'll see you at CRS, hopefully. So at this point, I'd like to say thanks for your time and for your interest, and I'd be happy to answer any questions.
Thank you very much, Dr. Frekin. Ladies and gentlemen, there are questions that you've typed in, so we'll get to those. But if you'd like to ask them in real time, you can raise your hand, we'll unmute you, and you can ask the question directly to Dr. Frekin. Thank you. And perhaps, Lou, if you want to walk through the Q&A that's been submitted. Yep. I believe there is one hand up. So we'll unmute you. And if you could identify yourself, please, and ask your question, Stefan.
Yeah, hello, thanks for this nice presentation. Just a question about the components you use for fluorescence detection. Is it avalanche photodiode? Is it photomultiplier? Is it something different? Is it single photon avalanche photodiode?
We're using APDs, yeah.
APD, okay. Okay. Can I have some other question, or is it?
Sure.
Yeah, sure. Yeah. What is the? It's not technical. It's just regarding the price of a cartridge.
Oh yeah, cartridges are $12 each. Yeah.
How much? Sorry.
$12.
$12.
Yeah,
okay.
And our customers love them because every time they're measuring one sample, they're getting the next one ready, and they don't have to do any cleaning in between the samples, for example.
Okay. Do you try to analyze samples like plasma or something like this?
Yeah, our customers use the technology for a whole bunch of different applications, but plasma can be run directly. There are papers about that in our library on our website. You can go see.
Okay. Okay.
And cell culture media.
You have no issue with clogging with plasma.
Right, because there are filters upstream inside the cartridge, right, that exclude the large particles. So we have people running biofluids directly on the cartridge in many different applications. Okay.
And just last question, sorry. What is the typical time for one acquisition?
Oh, good question. So measurements take a few minutes. That's it.
2 minutes.
Yeah,
okay.
Depends on the concentration, right? So because we're counting particles, if you have a very low concentration, then it takes longer to count sufficient particles to have the statistical certainty that you want. But typically measurements are, a few minutes long.
Okay. Just sorry, last one.
Problem.
Do you planify to have other fluorescence capture in the future?
Other fluorescence which?
Other fluorescence channel in the future?
More than 3, you mean?
Yeah.
Not right now. And part of the reason is because at this size scale, there's really not that many fluorochromes that you can put on a nanoparticle. There's a limit to how many antibodies can go on there. And so the major multiplexing that's happening for large scale flow cytometry is really not as relevant at the nanoscale. The more important measure is going to be sensitivity because of that same limitation, right?
Yeah, I understand. It's not a question of the number of different fluorochromes you can add on one single particle. It's the different subset of particles that could exist in one sample. For example, in plasma, for example, you can have 4, 5, 6, 7 subset different subset, and it could be interesting to have many detectors in fluorescence to finally analyze the EV subsets.
Yeah, okay, I understand. So for that, if we're not talking about co-localizing two different fluorochrome, or more than three fluorochromes on a particle, then it's perfectly fine to run the sample again with a different set of stains, right? Because it's a statistical measurement of the sample.
Okay. Thank you very much. Thank you.
Sure. Yeah, thank you for your questions.
I'll just jump in here. First of all, I wanted to also add on that last question about running in complex media. We just ran a sample for a prospect that was very interesting, where they had EVs and then they mixed the EVs in with plasma on purpose. And their goal is to be able to measure EVs in clinical samples where the EVs are actually less than 1% of the overall particles, and we were able to do that. We found about 1% when we measured that sample, so it was pretty cool. One of the questions from an anonymous attendee is typical size of AAV is 25 nanometers. Seems ARC has the limitation to detect AAVs. You're absolutely correct. Our current lower limit of detection is 50 nanometers. We're working actively on trying to do better than that, but right now we can't detect single AAVs. We've had some very good success with measuring aggregation of AAVs, if that's of interest. And the other question
I'll just add to that AAV question. If you have samples of AAV that you would like to share with us, we'd be interested in, we're always interested in trying new stuff. So if that application is of interest, it'd be cool to see if we could detect them fluorescently without the MRPS signal also. So please reach out to us if that's something that you'd like to see too. I'd love to try and do that with you.
Good. And then there was another question from Shiva. Hi, Shiva. She asks, how does this quantification compare with FFF mals based quantification or batch quantification with ribogreen or other similar techniques? Kind of a two-part question. You want to take that?
Sure. So we haven't done any direct quantification with FFF mals that I'm aware of. I know that there was a paper a long time ago from JHU and NIST that compared the size distributions obtained by MRPS with AF4. That's a little bit different. And then in terms of quantification with, how does it compare with the ribogreen? Well, so, and they're kind of orthogonal measurements, honestly, right? So we measure, so the ribogreen assay measures how much free RNA is left in solution after encapsulating into the LNPs, right? But it doesn't tell you anything about the distribution of that RNA among the particles. And so, for example, in some crazy world, you could say, well, half of the RNA got taken up into the particles, right? We detect half by the ribogreen assay still, but it could go into a single big particle, right? And the ribogreen assay won't tell you that. So obviously that's not happening, but my point is just that our technique tells you a lot more detailed information than the ribogreen assay can about the encapsulation and how it gets incorporated into the particles as a function of size, which is ultimately what we need to know, right?
Yeah, I think to add to that, I mean, one of the things that might be interesting to look at that we haven't had a chance to, but I know that some people making LNPs, for instance, they do the ribogreen assay to figure out the free, and then they also take the final sample and lyse it and do another ribogreen assay to try and see, how much is freed up. That would be an interesting one to see, how well they correlate. They should correlate, but it's not something we've actually done. And the other, I guess, point to make on FFF mals is that That's kind of a multi-step technique, and takes a lot longer, and so it's so it's and at the end of the day, you're still using basically DLS to quantify the particle size, which is why you have to use FFF first, because DLS only reports an average. So it's a less direct technique, I guess, would be my comment there.
Thanks for the great questions.
Ladies and gentlemen, any further questions? You could raise your hands.
Oh, I'm seeing more here. Sorry, I didn't see all these below. I needed to scroll. From Daniela. Hi, Daniela. Oh, AVs. Okay, same question. Okay. Leandra asked, have you tried multiple antibodies from different companies? Is there one that is better than the other? I'll let you handle that.
Sure. I mean, we try a variety of antibodies, some that our customers provide with their samples. So I'll put out there that, we're happy to measure samples as a demonstration. So if that's of interest to you, send us some, reach out to us, and we'll be happy to discuss that. I'd be happy to, share what antibodies that have worked for us also, but I'd have to look them up. So let's talk about that offline. Please send us an email about that. That'd be great.
And then I also see one here from Dennis. Hi, Dennis. Does the instrument require high ionic strength and addition of tween in the diluent for particle detection and characterization? We have had issues with the compatibility of tween with LNP stability. Good question. Dennis, the ARC is basically identical to the NCS1 you already have. So the detection via MRPS is identical to it. So you can think of ARC as a cross between an NCS1 and a flow cytometer, and that's a very oversimplified answer. As far as LNP stability with tween, we do have other surfactants that we've tried that work. Most people haven't had a problem with tween. It really depends on the composition of the LNPs you're making. So if you're having issues, I would recommend getting in touch with us. We have, for instance, we've gone to default on a lot of these samples now, where we're using poloxamer 188 instead of tween, because it's much more gentle and doesn't cause any lysing or anything of that sort. And for those of you who aren't aware of what I'm talking about, when we go into the smaller cartridges, we sometimes have to add a little bit of surfactant just to get the cartridge to wet. And our default is tween. But we've, as I said, we've found other surfactants. So if that's an issue, we can work with you to figure that out.
Any further questions, ladies and gentlemen? If you'd like to ask, either by Q&A or in real time, this is the time to do so. If nothing else, we'd like to thank Dr. Frecken for a great talk, very engaging as always. And I believe the Spectradyne team will be reaching out with a copy of the recording and such in due time. So thank you
very much, everybody.
Thank you. Goodbye.
Bye. Thank you.